Hello and welcome to the Skeletal Muscle Anatomy and Physiology lecture. We will mostly be talking about the physiology, so the function of skeletal muscle tissue, as well as just kind of microanatomy of muscles. So first and foremost, let's talk a little bit about the terminology I need you to know. When it talks about cells, we are talking about muscle fibers, and you want to look for terms such as myo, mice, and sarco. Any of those terms before other terms is going to imply that we're talking about muscles. So there are three types of muscle tissue. We have talked about these ever so slightly before when we were talking about histology. Today we're going to mostly talk about skeletal, which is going to be striated and you control it, so it's voluntary. The other two types of muscle tissue are going to be involuntary. Cardiac is striated, smooth is not, and cardiac is going to be distinguished by its intercalated discs, and it is going to be branched. And here are some nice graphics showing the three types of muscle tissue. And then these are nice slides showing the three types of muscle tissue, but from an actual slide. And you can see where the skeletal muscle tissue are nice and striated, so our nice lines. And you can see that cardiac muscle tissue is going to very much so kind of imitate that, but we have this branching going on in our cardiac muscle tissue, as well as we have these little lines or these discs known as intercalated discs. And then of course with smooth muscle tissue, it just looks very smooth because it is not striated. So let's talk about the main functions of muscle tissue. First is body movement. So the contractions are going to help muscles move bones. So muscles will be attached to your bones, which we have already discussed. Bones and kind of the weird features that can be associated with bones, so those bone features, which usually have something to do with muscle attachment. We are going to serve the purpose of maintaining posture. So contractions will stabilize joints and help maintain body positions, such as sitting upright or even standing. Laying down, not so much, but you still maintain tone in your muscles as long as you're alive. So it does do that. We are going to support soft tissue, guard body entrances and exits. So what I want you to think of with this is going to be sphincters. And we will talk about them a little bit more when we talk about the digestive system, but that's more of a smooth muscle function than anything. Producing heat, which is the process of thermogenesis, so it's contractions of muscles will generate heat. You can also think of shivering in the cold as being an example of thermogenesis, kind of a really intense, exaggerated one, but that does happen. And a provision of nutrient reserves, so amino acids can be used. So let's talk about some of the gross anatomy. So we have connective tissue components that I want you to know. So I do want you to know that surrounding everything is going to be the epimysium. So that is this term right here. It's going to surround the muscle. We have the perimysium, which is going to surround fascicles. You have the endomysium, which is going to surround muscle fibers. And then, of course, we have the tendons and aponeurosis. So here we're going to have a muscle straight off our model right here, a little cross section. We are going to have that cross section turn towards us with this little graphic here. So what we can see is that the entire muscle that we're looking at is surrounded by the epimysium. And then this muscle is made up of multiple muscle fascicles. Surrounding all of these fascicles, we have the endomysium. From there, surrounding each individual fascicle, we have the perimysium. Now you can also see that the endomysium is going to be kind of in between these fascicles as well. So composing of each fascicle, we have multiple muscle fibers. Surrounding those muscle fibers, we also are going to have that endomysium, which is kind of between each one. And you can see up here that it's kind of pointing to it, surrounding there. From there, the muscle fiber is going to be made up of myofibrils. So this just summarizes what I just said. I like color coordination. It makes me happy. So hopefully this helps you with that, and I would know this pretty well. So let's talk about the microscopic anatomy next. So we do have these little multinucleated cells. So they are multinucleated because they are multiple myoblasts that fuse together during development, and we will talk about what myoblasts are on the next slide. But we have these little myoblasts which are kind of squishing all together here, which is going to make an immature muscle fiber. So remember, that's just like basically a muscle cell. So it is multinucleated. You see all these nuclei here. And then you have this little myoblast that is kind of just hanging out. It didn't get clumped in with everybody else. And this is going to be known as a satellite cell. So here we just have that process, very nice and neat, and it shows us what we get in the end with our muscle fiber. Now what I want you to know is myoblasts are going to be muscle formative cells. So myoblasts will fuse to form multinucleated cells, which we said before, during the embryonic stage. So when you are an embryo. Myosatellite cells are going to be unfused myoblasts, and after an injury, these cells will enlarge, divide, and fuse with damaged muscle fibers to help with repairs. So do know the difference between myoblast and myosatellite cells. Myosatellite cells are more like support than anything. So each myofibril is going to further divide into the functional unit of muscles, and that is the sarcomere. So myofibrils will consist of bundles of protein filaments called myofilaments, and that is going to be what makes up the sarcomere. So shown below is going to be a muscle fiber with a myofibril extended out. So this little guy right here. And then if we look even further from this little zigzaggy line to another zigzaggy line that we can't see over here is a sarcomere. So let's talk about sort of the various anatomy that goes with a sarcomere. First and foremost is our sarcolemma. This is going to be the plasma membrane of a muscle cell, so that's going to be this kind of blanketing structure coming up and around here. That is the sarcolemma. Next, we have the sarcoplasmic reticulum. The sarcoplasmic reticulum, at least on this graphic, is going to be all the blue webbing. So it's very similar to smooth ER, and one of the most important things I want you to get from this is that its purpose is calcium storage sacs in the terminal cisternae. So the calcium is very, very important. It's 100% necessary for contractions, which, as we know, muscles serve the purpose of contracting to create movement of some kind. So that is incredibly important. Calcium is incredibly important in this case. So the terminal cisternae are going to be these kind of bulging sections here that surround this middle tube right here. So you have two terminal cisternae kind of bookending the transverse tubule. And specifically the transverse tubule, this little middle guy, is just a continuation of the sarcolemma. So the sarcolemma being this big blanket that is just connecting with the T-tubule. Now I do want you to know that the two terminal cisternae as well as the T-tubule here in the middle all together make something called a triad. Moving on a little bit further, if we take off the sarcolemma and the sarcoplasmic reticulum, this is what we have left. So we have our sarcomere, and keep in mind that you're going to have tons of sarcomeres lined up together to create the myofibril. A sarcomere is going to be made up of a dark A band and a lighter I band. So they alternate back and forth, and you can see down here in this graphic the I band being a little darker and the A band being darker and the I band being this lighter portion here. The A band is going to be made up of thick filaments, and we'll talk about what those filaments are here in just a second. I don't want to give it away, spoiler. And in the center here in blue or purple, however your color blindness might see it, it is creating something called the H band. This H band is just going to be from here to here. You'll notice further over right here that you have this overlapping of the I band filaments, the thin filaments, overlapping into where you have those thick filaments from the A band. So the H band is going to stop just before there. And you'll notice that if you kind of turn this on its side, so this, it kind of makes H's that are sort of lined up next to each other. If you can use your imagination, it looks like an H. The I band is going to contain these zigzaggy lines down the middle of it, and that is called the Z line, and it is a protein. So from one Z line to the next is one sarcomere. And then last but not least, we do have our little M line going down the middle of our A band, which is directly in the middle of our H band, which is part of our A band, right? So it's just that middle section of our H's. So speaking of our thick filaments, and keep in mind that this is our A band, is what we're talking about. It is made up of 200 to 500 myosin molecules. So just to give you kind of an idea, what this graphic is showing us right here is that very middle section of the A band. So this is the H's that are lined up, and this is the M line. So we're looking at the M line right here down the middle. And you have these little myosin tails with this little head here attached to it, and we'll talk about kind of what it does. So these heads are directed outward in a spiral array around bundled tails. So there's a ton of these little myosin tails with their little myosin heads sticking up. There's a reason for that, and we'll see it in just a second. So this is our thin filament. Keep in mind our I band. It's a really ugly I apologize. This is our I band, and it is composed of two strands of filamentous actin. So A bands are made of myosin. Thin filaments that compose of the I band are actin, and it is going to have grooves that hold something called tropomyosin. Tropomyosin molecules are going to block the active sites. So these active sites are going to be where we want to have the actin bind with myosin. Tropomyosin is sort of like a guard dog, if you will. It needs to be lured away with calcium in order for the active sites to become open. So once that happens, our actin can bind with our myosin. Now what happens when we have that binding is we're going to have a contraction. Those myosin heads are going to pull the actin forward, and they're going to contract. So when I say contract, that means that the sarcomere is not going to change length per se, but it's going to become kind of squished together. So if we have that little area of overlap, let me get my fingers on the screen, when we contract, these filaments are going to move closer together, and then when they relax, they're going to move apart. So you'll notice that the filaments do not change length during the contraction. The Z discs are just going to move closer together during a contraction and then move apart once they are relaxed. And that's going to happen once we have those active sites free of tropomyosin, and then we can have that binding of actin and myosin. Something kind of interesting and something that we can kind of apply to what we know now is rigor mortis. The idea of rigor mortis. Well, it's not really an idea, it's something that happens. So if you don't know, rigor mortis is the stiffening of the body 3 to 4 hours after death. It will peak around 12 hours after death and will diminish sometime around 48 hours. At least it will completely diminish around 48 hours. So what happens is that the sarcoplasmic reticulum, and if you remember, it's going to have a lot of calcium, as it deteriorates after you die, it's going to release calcium. And remember that calcium is going to lure away our tropomyosin. Remember, our guard dog. Once that tropomyosin has been removed due to that calcium, we're going to have a contraction. So what happens when the muscle contracts is it's going to stay contracted until you have ATP that allows for that contraction to stop. If you don't have ATP, the muscle won't relax. So you're dead, you're not going to produce any more ATP. So that is why your muscles contract and it's hard to move a body after it has died. So after that sarcoplasmic reticulum is deteriorating, all that calcium is being released, we have that tight contraction happening, all the limbs are stiff, the body is stiff, can't relax because there's no ATP. But what ends up happening is that the myofilaments will start to decay, and that will cause that kind of relaxation. More so you're just decomposing more than anything, and that will cause the body to unstiffen. So next up, we're going to talk a little bit about the neuromuscular junction. This is the region of contact between the motor neuron and muscle fibers. So we are going to talk about the nervous system, I believe, in the next lecture. So this is a good sort of warm-up for us, if you will. So I do want you to know this term excitability. Like neurons, muscle cells are going to generate and respond to electrical signals. So that is how neurons talk to each other, is through electrical signals, and we'll talk about that more in the next section. But do know that this is called action potentials, and we'll talk about this more when we talk about the nervous system. But just know in general that that's what we're looking at. So here we have little axons. So this is going to be the talking piece of a neuron. The axon is going to send signals and this is where it is meeting a muscle fiber, and it's meeting it at the motor end plate. This is more of that. It kind of looks like worms or parasites, but that's just axons. So I do want you to know a few of these terms here when we're talking about our motor end plate. This is what it looks like. What we have here is going to be the axon. And then of course down here, this is going to be the muscle fiber. And this is where it's coming into contact with each other. So a synapse is going to be the region where the nerve fiber makes contact with its target cell. This could be any cell, but in this case, it's going to be muscle. And then we have something called a neurotransmitter. What I want you to know is that this is a chemical. The release is going to cause the stimulation of a cell. This can be kind of any cell, but again, in this case, it's going to be a muscle cell. And specifically, the neurotransmitter we're going to talk about right now is going to be acetylcholine, or ACH. Next, we're going to talk about the synaptic terminal. It's going to contain vesicles, which we've talked about vesicles in one of the first lectures when we talked about cells. So if you need to review that, that's fine. But this is going to contain vesicles with acetylcholine in it. So these are the vesicles that we are talking about. That is the synaptic terminal as well. This structure down here. The motor end plate is going to be where we have acetylcholine receptors. So that's going to be down here. So this is just waiting to receive the contents, which are the neurotransmitters, from these vesicles. And then we have this term acetylcholine esterase, and I believe I'm going to talk about it here in just a minute, but just know that it's an enzyme that breaks down acetylcholine in order to relax. So here we have the synaptic cleft, which is going to be this space between the neuron and the muscle fiber. I really like to think of this space as being kind of like if you've ever had a younger sibling, or maybe an annoying cousin, or I don't know, something like that. It's the I'm not touching you type of space. They're not actually touching at all. There is just this space in between, but it is very, very small. And then of course, we've got our acetylcholine esterase, which we've just talked about. I do want to talk about different types of contractions now that we've talked about how muscles do contract with those sarcomeres just kind of squanching together, as well as how we get a message through to say to contract. We kind of understand those concepts a little bit. We're not going to go into them too much as I feel it's unnecessary to do so. But we do have two types of contractions, and one has two subcategories. So we have isometric muscle contraction, which means that we're going to develop tension, but not change length of the muscle. So this is specifically talking about within the muscle. So you can see in this first picture that it is showing isometric contraction. There's tension, but we're not shortening the muscle. There's no movement going on. So this would be like if you were pushing against a wall, and unless you're the Hulk, you're not going to push through that wall, but you're going to have a lot of tension going on, and even though you are pushing, you're not going anywhere, nothing's moving, the muscles in your arm are not changing length, but there's still tension. Isotonic muscle contraction is going to be where you do have shortening or lengthening as well as tension development. So the terms for that is concentric isotonic muscle contraction, and that is where you have tension develop and the muscle is shortening. So picking something up is a good way to think of that. And then eccentric isotonic muscle contraction is going to be tension developing while the muscle is lengthening. So setting something down would be a good example of that. And then this picture just shows more of the same. I like to give a little bit more than just that last side to help you visualize these types of muscle contraction. And very importantly, we need to talk about energy production in muscles. So the contraction of muscles require a large amount of energy, which we are going to get in the form of ATP. So we talked about ATP, and we know that it comes from mitochondria. So mitochondria, powerhouse of the cell, they're kind of the powerhouse of more than just the cell. They're the powerhouse of your body in a way. They produce all the ATP. It's very important. And ATP is present only long enough to power a contraction for a few seconds. But that's very important for those like minor contractions and whatnot. If you are experiencing something tragic, in my opinion, strenuous exercise, as you can tell, I obviously don't do that. But if you do, or have in the past, you're going to need a lot more ATP to be made in order to exercise continuously. So if you're doing something kind of quick that doesn't require too much energy, you just use up the ATP that's there. If you go past that point, you're going to have the need for more ATP to be made. So let's talk a little bit about the sources of ATP. First is creatinine phosphate system. We're not going to talk about this too much. What I want to get at more with you guys is the idea of glycolysis, where you have limited ATP, and aerobic metabolism, where you're going to have more ATP. Specifically, what I want to talk about with these systems is this: the presence or absence of oxygen. So glycolysis, there is no oxygen present, but you're going to have a lactic acid buildup. When it comes to that lactic acid buildup, that's where you feel the burn. So you get this lactic acid buildup in your muscles, and that's that kind of burning, warm, uncomfortable sensation that you can get when you do a sprint or a lot of excessive energy, exercise, but that is that burn. Aerobic metabolism is going to require oxygen, hence the name aerobic, meaning, air. And it's going to produce water and carbon dioxide. So it's going to produce water and a waste. Fatigue, I do want you to know the definition of this, is progressive weakness and loss of contractility. So contractility meaning your ability to contract your muscles. Some causes of fatigue, aside from being a teacher, I'm kidding guys, I love it, is ATP synthesis declining as glycogen is consumed, having an ATP shortage, the buildup of lactic acid causing your pH to lower, or leaky calcium ions. Very quickly, we will talk about oxygen debt. So this is the need to breathe heavily after strenuous exercise. So again, if you've ever exercised or have done something that is strenuous in nature, you notice that you breathe a lot heavier. You might start to pant in a way, not unlike a dog, or maybe you breathe in deeper. So the purpose of this is to replace your oxygen reserves. So you're going to have oxygen in your myoglobin and hemoglobin and dissolved in your plasma, so that's in your blood. Now that gets used up as your muscles require the need for more oxygen. Remember that aerobic metabolism is going to require oxygen in order to make the product of water and carbon dioxide. So you're using up your oxygen as you are exercising, and you need to replenish that, right? And that will be almost exclusively in your blood and your muscles. You are going to need to replenish your creatinine phosphate system as well, and you also need oxygen to reconvert lactic acid to glucose. So once that lactic acid is built up in your muscles, you got to find a way to get rid of it. So it just can't be like flushing a toilet and just letting it go. It needs to be converted into glucose, which is, sugar. And then I believe last but not least, we're going to talk about a few conditions that affect muscles. The first being muscular dystrophy. There are several different types of muscular dystrophies. You can have genetic, and then producing muscular weakness and deterioration. So if you've ever been bedbound for a long period of time, perhaps you've broken a bone in your leg, or you were ill for quite some time and you had to stay in bed for a while, you may notice that your muscles start to weaken as well as kind of deteriorate. So if you were quite muscular and then you got bedbound, you might notice that your muscles will have shrunk in a way. That is just a type of muscular dystrophy. Duchenne-Becker muscular dystrophy is actually a genetic, inherited, sex-linked type of muscular dystrophy. It is the most common form and best understood form. It appears in childhood and generally will only affect males. 1 in 3,500 to 5,000 newborn males worldwide will be affected by this, and unfortunately most individuals die from this disease due to respiratory paralysis. Because this is causing the deterioration of muscles, the muscle that causes you to be able to breathe, the diaphragm, which is going to be directly underneath your lungs, ends up being affected by this disease, and unfortunately that is the cause of death. The next condition we're going to talk about is tetanus. Don't get this confused with tinnitus. Tinnitus is something that happens in your ear. Tetanus is something that occurs from a toxin in a bacteria, Clostridium tetani, that suppresses the mechanism inhibiting the motor neuron activity. So this bacteria is going to thrive in low oxygen areas like deep punctured tissues. So one of the most common ways to get tetanus is from rusty nails. I remember my parents always being very, very aware of where I was playing outside, and especially if it was kind of like older, like play equipment or kind of like playing in the backyard. I could never run around with no shoes on for fear of stepping on a nail or a rusty nail or something that may have this bacteria on it. Once it's in your system, it will result in a sustained and powerful contraction that will affect the muscle that it punctures. Severe tinnitus, tetanus, see, I did it, can have 40 to 60% mortality rate, but deaths are rare in the United States due to most people being immunized for tetanus. And then I believe last but not least we have ALS, which is amyotrophic lateral sclerosis, and this is a progressive neurodegenerative disease which affects nerve cells that supply muscles. And since muscle does not have the electrical signal, you can't maintain muscle tone, and that muscle will start to atrophy. Remember I said at the beginning of this lecture that as long as you're alive, you have muscle tone. And that is because you're going to have those electrical signals going to your muscles saying, hey, I'm alive, keep having that tone, you know? But if there's no electrical signal, such as with ALS, or if you die, then that muscle tone can't be maintained and the muscle will start to atrophy. There are two types of ALS, sporadic, which is 90 to 95% of the cases, and familial, which is obviously less, and it is inherited. I believe this is the last slide, it should be, but it's kind of a fun topic, and that is pickle juice good for cramps? The answer is yes, and that is because of the sodium content of pickle juice through vinegar. So if you know anything about pickle juice is mostly vinegar, and that sodium is going to help relax the cramping muscles. From personal experience, pickle juice and an ibuprofen are some of the best ways to treat menstrual cramping. I've also heard from quite a few football players that their coaches will have bottles of pickle juice for them on the sidelines, or other sports, I suppose, would probably have that as well. But it's good for muscle cramps in the legs and whatnot as well. The one thing I can say with certainty is Gatorade is not good for cramps. It's actually not good for you really at all, to be fair. I'd say probably the no sugar Gatorade is fantastic, but regular Gatorade, as delicious as it tastes, is because it has a lot of sugar in it. Electrolytes in that amount is honestly not that great for you unless you are an extremely sporty person and you're exercising quite a lot and need those electrolytes replenished. Otherwise, I'd say stick to the sugar-free Gatorade. This is the last little topic here, and that is the other two types of muscle tissue. We're going to start with smooth muscle, and it is just one slide each because we will talk about these a little bit more later in our lectures. So, smooth muscle is going to be composed of fusiform cells that contain only one nucleus, so they are not multinucleated like skeletal muscle tissue. There are no striations, no sarcomeres, or Z-discs. The filaments are scattered throughout the sarcoplasm, and they have a reduced sarcoplasmic reticulum and no T-tubules. The calcium is going to be coming from extracellular fluid, and it's slow but steady contractions. It is going to go through something called peristalsis, and we will talk about peristalsis when we talk about the digestive system, but that is a process in which food, as well as food after it's been digested, is moved through the digestive system. And it's a process of, if you pretend that my hands are making up a tube, it's going to be a contraction behind the bowl of food, and then a relaxation in front of the bowl of food. So then that food moves down, and then it just rinse and repeat. Contraction, relaxation. Over and over until we've moved something through the digestive system. And here we are, last but not least, cardiac muscle tissue. So the cells are going to be shorter, thicker, and linked to each other by intercalated discs. Do know that this is where we are going to have electrical gap junctions that allow cells to stimulate their neighbors. We do have a term called autoarrhythmic, which is going to be from our pacemaker cells located within the cardiac muscle tissue. And as the name implies, a pacemaker cell is going to cause those contractions that cause your heart to beat. And we'll talk about that more when we talk about the heart later on in the semester. It is going to use aerobic respiration almost exclusively. It is going to contain more mitochondria than the other two types of muscle tissue, and it is resistant to fatigue. And that is a very good and important thing to make note of. If your heart was to experience fatigue, you would die. Your heart has to beat constantly, and you don't control it. So if it did get tired, that wouldn't be a good thing. But there are ways to make your heart tired, such as blood doping. Blood doping is going to put more blood into your body than is necessary at the time, and that is going to cause the heart to work harder than it needs to, and that can cause heart failure or fatigue, essentially, for lack of a better term. The heart muscle, cardiac muscle, can also use lactic acid and is very beneficial during exercise. So some of the best exercise you can do is cardio, as the name implies, that is going to help your heart. And I believe that is it for this lecture. If you have any questions, don't hesitate to ask.